From Diamond Light Source, this is the Diamond Podcast.
Hello and welcome to the July edition of the Diamond Light Source podcast. This month we're on location at the Light Source to bring you inside access as we attend one of the facility's Inside Diamond open days. We'll be meeting some of Diamond's top engineers and technicians who designed the components of the synchrotron and keep it running smoothly, hear from Diamond's CEO Gert Martelijk about the main highlights of open days such as these, and talk to a scientist working on one of Diamond's latest beamlines, I-24, that's enabling research that wasn't possible before.
Typically other beamlines would have an X-ray beam of maybe 50 to 100 microns, whereas on I-24 we're now working towards 2 or 3 microns. If we can reduce the beam, the very weak signal becomes visible when otherwise it wouldn't have been.
Beamline scientist Danny Axford will be explaining what insight can be given by such small beams of light later in the show, when we also hear how Danny's beamline has been used in the battle to develop more effective antihistamines, as well as hear what the visitors at Inside Diamond Day have to say about science at the synchrotron. So all that coming up in this month's Diamond Light Source podcast with me, Mira Senthilingam.
The Diamond Podcast. For more information, look us up online at diamond.ac.uk/podcast.
So this month, we're on site at Diamond, bringing you an insight into the Inside Diamond Open Days held at the facility, a day where members of the public get to come along and tour the light source to learn the workings of a synchrotron.
There are many talks and tours throughout the day, but before they all began, I spoke to Diamond's CEO Gert Martelijk about the aims of getting the public inside Diamond.
We are living within a community, so we need to, from my point of view, show the community, invite the community to see what we are doing, really. To give them a feeling, on one side, how interesting science is. And the other is the aspect that I think we are for 80% financed by the government. I think it's always good for the people to see what happens with their taxpayers' money. Where does it go? What is it about? We really do something. Or we can help people, for example, in terms of, we think about spectrum of things, to look at viruses, virus structure, to look at histamine structure. On the other side, look at conservation of the Mary Rose, or at hip replacement, and so on. It's a broad spectrum. Well, there's basic science to understand functioning of life and to understand magnetism on the other side.
And what does a day involve?
Well, a group comes here in the morning or in the afternoon, and they will first have a talk, introduction into an overview of Diamond. It's the managerial structure, but also the broad science structure. And then for an hour we take them around and show them the accelerators. We show them the instrumentation, the beamlines, and we tell them about a few science examples, and hopefully they have an understanding how broad it is.
Diamond's chief executive, Gert Martelijk. Now, the open day to go inside Diamond starts with a talk explaining the workings of an electron accelerator like Diamond, and how different beams of light can be produced by basically getting electrons to move close to the speed of light around a ring. Following this talk is access into the ring itself to see where it all takes place, and this is guided by the many engineers and technicians working at Diamond, providing their insight into running such a large-scale yet precise experiment.
As all the physicists will know, electrons carry on in a straight line and you can bend them with a magnet. So we generate...
Hello, my name's David Hawkins. I'm a mechanical design engineer at Diamond Light Source, and today I'm a tour guide showing people around the facility.
So, David, what are the key points that you kind of want to get across to visitors as you take them around the facility?
A general appreciation of how the machine works, and then looking at individual pieces of the machine, and then taking them to a talk by a scientist so they can understand what the scientists are trying to get out of the machine. What I do try and do is to make it understandable. For instance, the radio frequency cavities, I try and make analogous to the kitchen with the microwave unit.
And what's the general reception of your kind of crowds that you get, of the groups that you take around?
I find they're usually aghast, and also the sorts of work that's done. Things like working on the hip joints, the Mary Rose Trust, being able to read the deep sea scrolls without unrolling them. It just carries on and on and on. They really are amazed.
And what about your role here at Diamond then? So you're a design engineer. What do you design, I guess, and what does it involve?
I design bits of the beamline. I've designed things like diamond windows, diamond fluoresces when it's hit by light, and we use that to work out where the beam is. So as the beam comes into the hutch, we want to know where it is. But designing a piece of diamond 0.9mm thick that's got to take a lot of heat without breaking, that takes a bit of effort.
And so what's the role of this diamond window with regard to the synchrotron as a whole, and how important is it?
When you're commissioning that beam, you need to know where the beam is because it can get deflected all over the place. So we had 4 diamond windows down the length of that particular beam line. I-15, it's the high pressure extreme conditions beam line. So we had 4 diamond windows down there. So as we were getting the light into the hutch, we could find out where it had gone wrong, and they were invaluable on setting up. Otherwise you were just working in the dark. You had a big dark room and you didn't know where the beam was.
So they help you guide where to get the beam to, I guess, and then do they stay in place or are they then removed?
They stay in place all the time. The second role is they provide a barrier, so if you have a loss of vacuum, you don't contaminate the next section up.
I'm Alan Morgan, and I'm one of the beam diagnostic physicists here at Diamond.
So what do you, I guess, diagnose here? So what's your day-to-day role at Diamond?
Well, I'm part of the diagnostic team on the machine side, so we're looking mainly at the electrons. So we have systems to find out what the position of the electron beam is in the machine, because we need to keep that very stable so that the beam lines have a stable thing to look at and use, and also the amount of charge in the beam, basically how well behaved it is, and also if something's going wrong, to find out what's going wrong with it.
So is this in every beam line, so what's going on?
Mainly it's on the machine side. We do have position monitors in the beam lines, and the beam lines do have some diagnostics, but it varies from beam line to beam line. Some have a lot, some have very few.
And how, I guess, do you maintain stability? So what do you have to look into, and what do you have to maintain?
Generally, you have feedback systems to keep the stability that we need. So we look at where for say position, we look at where it is, and we look at where we think it should be, and we try and move it to where it should be. For us, feedback systems are a vital part of keeping it stable enough.
And I guess, what are the actual requirements? So how stable does the beam have to be, and how important is it that it stays stable at this particular level, say?
Generally, with our feedback systems, we can keep it stable to within about 200 nanometers of movement. Some beam lines are sensitive to motion more than others. It really depends on what experiment they want. So there are certain beam lines we will be more aware of that they are more sensitive, and we'll check with them if they can see things. They're almost a bit like the canary. If they see something, then we need to check it. If they can't see anything, then probably nobody can.
My name's Linda Pratt. I'm a software systems engineer, and I work in the controls department.
So what, I guess, do you have to control as part of the synchrotron, and what do you kind of look into?
We have different technical areas of control. The 2 of the major ones are motors and the vacuum systems. We also have diagnostic systems and other instrumentation to monitor the state of the machine or beamline, or indeed part of running the end stations for the experiments.
So that's quite a few things there. Could you pick perhaps 1 or 2 and explain how you control them, or how they're controlled, and how important it is for them to be controlled?
The vacuum systems, for instance, a typical vacuum component like a pump will have a specialist controller piece of instrumentation, and then we interface that into one of our computers, either a standard PC or a specialist rack-mounted computer, and then that ties into the monitoring alarm system and the screens that the operators use. So the operators can tell, and the experimenters can tell from their computer screens, whether a particular pump is on or off, If you add all that up together for controlling the vacuum system, it maintains the quality of the vacuum that allows the beam to travel around the storage ring or down the beam lines.
So I'm Joe Williams. I'm a mechanical design engineer at Diamond.
What aspects do you design and engineer?
It's to do with the beam lines. So outside of the storage ring wall is when you get into the beam lines. So we design the equipment that goes on the beam lines to make sure it's tailored specifically for the sort of science the scientist wants to do on that beam line. Most of the work is on the end stations. Most of the end stations are specific to each beam line. So we're tailoring the design
to adjust the kind of the type of the beamline accordingly to what's going to be looked at, or
it's more handling different samples. On I-24, they're looking at microscopic things. You're looking at some of the other beamlines like Jeep. They're looking at much bigger things like aircraft turbine blades. So you've got a completely different mechanism you're going to need to handle that to holding a tiny little crystal.
So you deal with the actual components or the pieces of equipment that handle the samples or hold the samples for testing?
Yes, but it's all the equipment before that to tailor the X-rays to be specific for what that sample needs. First of all, you want to block out all the radiation that you don't want from the storage ring. So the first component is something called a collimator, which is a big block of lead, which blocks out all the radiation that you don't want. So you just get your X-ray beam coming down the centre of a tube. After that, you'll get things like monochromators, which then select the energy of the beam line. And you'll get focusing mirrors, slits, which are just blades that come in from the sides and the bottom and top, which define your wide beam into a nice little narrow beam.
So all of this variety of equipment basically homing it in, so from this large beam just narrowing it and focusing it and narrowing it and focusing
That's it, exactly, yes. And because, I mean, essentially that's what your scientist is interested in, is getting the right energy of X-rays at his sample.
So it's clearly not about building a synchrotron and then leaving it to run itself. Every step and every material along the way needs to be monitored closely by the wide range of highly skilled scientists and engineers at the light source.
Did you know that Diamond's floor area is 45,000 metres squared? That's almost the same as 8 St Paul's cathedrals.
This is the July edition of the Diamond Light Source podcast, and this month we're taking you inside Diamond and down into the storage ring to see what the public have access to when the facility holds its Inside Diamond open days. Now, we've heard that the day involves a tour around the synchrotron, so seeing where the electrons are generated, how they're boosted to reach speeds close to the speed of light, and how various beams of light can then be generated by the use of magnets. But along the route, it then makes sense to see one of the many beamlines and hear from the beamline scientist about the scientific research made possible by that beam of light.
So, welcome to Beamline I-24. So we're macromolecular crystal. My name is Danny Axford, and I'm a support scientist on Beamline I-24, which is a microfocus macromolecular crystallography.
What kind of research is done here? What can be looked into here?
Primarily, we're looking at the atomic structure of biological molecules. So these are molecules that are fundamental to how life processes work. And I-24 in particular has been designed to look at the most challenging samples that have so far proved difficult to analyse. So we use a specially highly focused X-ray beam which allows us to probe smaller samples. Typically we're analysing crystals, and the more complicated the molecule, the harder it is to produce a crystal. And when you do get a crystal, it's typically small. So we've designed our X-ray beam to be focused as small as possible, which helps in this analysis. The idea with macromolecular crystallography is, in order to get a strong enough signal from the diffraction experiment, you grow a crystal, and the crystal is an ordered lattice of the molecule. And the bigger the crystal is, the stronger the signal you'll get. Now, complicated molecules really don't like to grow into crystals. Typical examples are membrane proteins. Now, these are typically embedded in the surroundings of cells, so they're involved in transporting molecules in and out of cells. And because they sit in the membrane, then they're not water soluble. So to grow a crystal from solution is very tricky.
Why does this feature of them make them harder to get into crystal format then?
Because typically they would be surrounded by the lipid that forms the membrane. And we're not interested in the lipid, we just want to look at the protein, the membrane protein itself. But if you remove too much of the lipid, then these molecules just fall apart. What you have to do is replace the lipid with detergent, which allows you to solubilize these molecules and then grow them into crystals.
And they form quite small crystals?
Typically they will be very small, often very thin, so you often get plates, so they're sort of two-dimensional. And this lack of volume means that the signal that you can detect from these crystals is very, very weak.
So why has seeing such, say, small crystals of samples been difficult with other beamlines, and how has this beamline overcome that problem?
Okay, so typically other beamlines would have an X-ray beam of maybe 50 to 100 microns, whereas on I-24 we've managed to get our beam below 10 microns. We're now working towards 2 or 3 microns, and if your sample is only 2 or 3 microns and you hit it with a beam that's maybe 100 microns, then most of the X-ray beam will be missing the sample, and essentially that is just adding noise into the signal that you detect. So the signal that you're looking for is just washed away by the noise. Whereas if we can reduce the beam to the size of the crystal itself, then the signal to noise ratio is increased massively, and so the very weak signal becomes visible when otherwise it wouldn't have been. So we can actually have a look at the beamline and see the components in action.
So we've come through to the beamline now, and there's a large metal box, basically, in front of which there's another attachment out of which the actual beam, I think, the X-ray comes out. And then in front of that, there's a very small, minuscule looking pin onto which the sample is placed. So how small is this sample again?
Okay, so some of our samples are just a few microns in size. So we're talking less than a tenth of the size of a human hair. So even under the optical microscope that we've got integrated into the beamline, these samples are very difficult to make out.
And this large kind of metal box almost, but very thick metal box behind it, this is where I think this beamline comes into its own, and it's quite unique.
That's right. Inside this vessel here, we've got an extra set of focusing mirrors. So that allows beamline I-24 to get the really small microfocus beam that's required to hit these samples.
So that hits onto the sample. I mean, the pin which the sample's on is just a couple of centimetres in front of where the beam comes out, but then that's diffracted onto what looks like a big kind of, it's about maybe 0.5 metre squared board, about 1.5 metres away.
That's right, this is the detector. We can actually move it closer in. It's extremely sensitive and it can read out very quickly.
And well, you mentioned then that an example of quite a complicated molecule then is a membrane protein. But what other examples can you give? And I guess what has been looked at so far on this beamline?
Okay, so another good case are virus particles and samples, where these are very large molecules of the order of millions of atoms. Typically they won't freeze very well at all. So often, instead of freezing the samples, we have to collect the data at room temperature, and that means that the samples do not last for very long at all. They're destroyed by the X-rays inside of a second, which is why we need the sensitive detector that's very fast, and typically we'll have to get through lots of samples and then try and combine the information that we get from them into one complete picture.
So viruses, membrane proteins, so as you mentioned, largely biological molecules.
I mean, in some cases we will shoot small molecules which could be in the form of drugs, for example, but these would be attached to the biological molecules. So we can see where the drugs attach. That allows us to maybe tweak the design of the drugs to have a better interaction with the target molecule.
Danny Axford, beamline scientist at Diamond's I-24 microfocus macromolecular crystallography beamline. And as Danny concluded there, this beamline enables scientists to visualise how drugs are binding to certain proteins found in our bodies, such as membrane proteins. And this is what Dr. Simone Viand from Imperial College London has been doing. More specifically, she's been investigating the binding of antihistamines in our bodies.
Yes, so I work on different proteins, which are all membrane proteins, and the latest result is the histamine receptor. And the histamine H1 receptor is a protein which is specifically recognizing histamine, and histamine is a compound that is involved in our local immune response. So as part of our immune response to foreign objects, such like pollen or pet hair or whatever, food, for instance, histamine is produced in our body. Now, binding to the H1 receptor, histamine is able to trigger the inflammation response associated with the allergic reactions, such as hay fever, food and pet allergies.
So you were looking at the histamine H1 receptor. So whereabouts is this particular receptor found? And so what cells is it found on and what role does it play?
The receptor is found in all sorts of tissues, in the human tissues and, mammalian tissues essentially. So the histamine is actually a substance which is produced by our body. And we also have the receptor recognizing this histamine in our body. So in principle, an allergy is basically a hypersensitivity of our immune system.
And so things like hay fever and I guess even just allergies to particular things in the environment, that causes histamine production, which then comes and binds to this receptor. Is that right?
Exactly.
And so I guess you've been looking at the structure, though, of the actual receptor. And why is it important to know this structure? And what was, I guess, known about it before? And what have you now been able to see that's new?
It was not a lot known before about this receptor. Well, in biochemical terms, yes, but not how it looks like exactly. And for us, it was really important to have a very high-resolution picture of how this protein, this receptor, looks like. And especially what we show is this receptor binding an antihistaminic drug to it. What we actually could show is a high-resolution picture of this complex. And what it means essentially is that now the pharma companies can produce a highly specific drug which is treating or getting rid of the allergy symptoms, which has no side effects. Because drugs so far against allergies, they have a lot of side effects. These drugs are not really specific against only this receptor. For instance, heart receptors or proteins in the heart, which like the potassium channel, and therefore we can have problems with the heart like arrhythmia or dry mouth, drowsiness, sickness, all these kind of things.
So how do current, say, antihistamines work then? Do they bind to this histamine receptor and therefore just stop histamine binding to it?
Exactly. That is what happens. So the antihistaminic drugs, they bind to the receptor and block it from histamine binding to it.
Knowing then the structure of the receptor, how could you make then or potentially think about designing a drug that would perhaps attach to this receptor better? What can you now do knowing this structure and knowing what happens when the antihistamines bind?
Yes, because we have really the atomic detail of how this structure, the binding site, the active site of this protein reacting with histamine or drug molecules, how it looks like in the smallest detail, in atomic detail. The pharma industry can really specifically design a drug which is only binding to this receptor. We could really see the interactions, the specific interactions of the drug molecule doxepin, which is an antihistaminic drug available on the market. And by comparing this protein, this receptor, with other receptors of known structure, we then can also see what are the differences, what is important in this environment for the selectivity of the drug molecules, etc.
So you've been able to see, I guess, how antihistamines bind to the receptor. What would the next steps be to try and make a better drug, or to think about designing a better, more targeted drug?
For designing a highly selective drug, really, it is now over to the pharma companies. But what they will probably do is design a whole library of drugs which is only interacting with the residues of the active site of this receptor, then screen it probably in animal models to see, is it highly specific? Does it interact with other receptors, with other proteins? Do side effects still occur or not? And make it really specific enough and reduce it. For me, or for us, one of the next steps will probably be just to see and how different antihistaminic drugs which are available on the market bind to this receptor, to see what are the interactions there, what is the difference to the drug which is bound currently, and are there some interactions which we didn't see so far? Are there more interactions or less interactions? All these differences, they have a big influence on designing a highly selective drug against allergies.
Simone Viand from Imperial College London. Now this month, we've been hearing from tour guides, engineers and the chief executive of Diamond about the importance of letting members of the public inside the facility to give them a good insight into how a synchrotron works. And a wide range of visitors across all ages and professions do come along. But what do these visiting members of the public actually think of the facility?
My name's Thea Labadi and I'm in market research, so nothing to do with science or physics at all. And the reason I came along is because my flatmate's quite interested in sciences and everything else that goes around, and she said, would you mind coming along? There's something happening around work. So I said, no, that's fine. I'm very, very glad I did.
What were you expecting before you came along?
I was expecting something, I think, a bit more physics orientated. So a lot of mathematical formulas and just explaining what's happening, but more in a scientific way. Whereas what you've done today is explain what is happening, but in everyday language, which is very, very helpful.
And did it help, I guess, because you go on a tour and actually see the whole facility, so that must have been quite different?
Yeah, that definitely helped as well, because you saw what was happening in the presentation, but to actually see it real life was very different to what I imagined, even though I saw the picture in the presentation, to actually see how it was built and just all the different magnets that are around it. Fantastic.
And I'm Adam and I'm hoping to study at Leicester to do physics. I just can't comprehend how massive the whole place is and the amount of equipment and how technical it all is, and just like how technical sort of experiments on this scale are, and how much equipment's required and how precisely it has to be monitored, and everything has to be at the right temperature for it to work. Basically how technical, and it's just mind-blowing how technical the whole thing is.
Hi, I'm Liz. I'm a student nurse. I'm in my third year at Oxford Brookes University.
And so what did you think of the open day? Well, I guess what were you expecting before you even came along? Did you know much about Diamond before?
I didn't know anything really, apart from that it was an accelerator, so I didn't really know anything at all about what they did. And the talk was really good because it was scientific based, but it was really easy to understand. Obviously the medical side of it I was more interested in, and I think it's amazing what you do here, but yeah.
And was there anything in particular you found interesting? Like, was there anything in particular that's retained in your mind?
Yeah, obviously for me, the important things are like the cancer research that you're doing and the very topical thing about hip replacements. So for me, I'm going to go back to my placement that I'm in at the moment and tell them all the stuff that I've learned about that.
So it seems the sheer scale of the synchrotron, combined with the wide variety of scientific disciplines that are studied there, are what impressed the public the most. This is something that still impresses me, as each of our podcasts focus on a completely different area of research, ranging from microbiology and environmental pollution to reading the Dead Sea Scrolls and preserving ancient pieces of art. Now, unfortunately, we've come to the end of this month's podcast, but we'll be back in September with more research, news and events from Diamond. In the meantime, if you have any questions about Diamond or the research that you've heard about in this podcast, you can email podcast@diamond.ac.uk. And as you've heard so much about the Inside Diamond Open Days held at the synchrotron, you can also get details of the next Open Day online at diamond.ac.uk. Thank you very much to Gert Martelick, David Hawkins, Alan Morgan, Linda Pratt, Joe Williams, Danny Axford and Simone Viand for taking part in this month's podcast. I'm Mira Senthilingam, and thank you for listening.
The Diamond Podcast is brought to you by Diamond Light Source and produced by thenakedscientists.com. There's more information on our website at diamond.ac.uk/podcast.